So I'm gonna move forward now, and I'm excited to introduce our second
Thank you.
speaker, Dan Bettinger, who is the Senior Field Application Manager at Carterra, and with over 2 decades of experience in therapeutic monoclonal antibody generation and characterization. Dan now leads Carterra's global application science team and was instrumental in launching the LSA platform. And today, Dan will showcase how Carterra's HT-SPR technology is setting new standards in throughput and scalability, enabling transformative library screening where entire target backlogs can be screened simultaneously. And Dan, I see you're already sharing your screen. Please go ahead.
All right. Thank you very much for the introduction, and thank you for having us here today on this ACRO webinar. So I am going to talk a little bit about our discovery things. We changed this a little bit from that abstract in that the topic of this was supposed to be about quality and development. So I tried to focus a little bit more on that. And if I can advance my slides. Excuse me, I think the laser pointer is giving me a complexity here. Let me turn that off. Okay. Maybe I can turn it back on now that it's working. Yeah, okay. Sorry about that. Okay, so this is the Carterra ecosystem, as I like to call it. So Carterra sells high throughput SPR systems. We like to call it HT-SPR, using an array-based approach. We have 3 instruments: the original LSA, the more recent updated signal enhanced version, the LSA XT. And last year we launched a new instrument called the Carterra Ultra, which has some pretty significant changes to the optics and fluidics and thermal approaches to make it a more sensitive instrument for things like small molecule analysis, or maybe more challenging biologics. We have a software environment where we have our navigator software, which controls the instruments and collects data used to design the experiments, and 2 analytical software pieces. One is called kinetics and one is called epitope. Relatively self-explanatory what those focus on. And then we have a whole line of biosensor chips and consumables. So Carterra has been selling these platforms since the end of 2017. And we've now in more than 100 customers around much of the world. Many have multiple instruments. So we have a lot of instruments in North America, obviously. We have sites in Europe and many customers there. And in Asia, we have quite a few placements, and we've partnered with Revvity to be a regional distribution and support center there. So we have a reasonably good global coverage for that. So again, we have these array-based HT-SPRs. These have really become the go-to for high throughput characterization of these core applications that are used in drug discovery, being kinetics and affinity analysis, competition-based epitope binning, peptide or mutant mapping, or really sort of matrix, many binding on many type interactions, and quantitation assays. So in addition to having a hardware architecture that makes these assays really high throughput and not use very much sample at all, we've put a huge amount of effort into our analytical software packages to make them sort of rapid, easy to use, but also give lots of very visually rich information, including sort of our proprietary network plots and heat map visualizations in the epitope software. So, as I mentioned, this is becoming sort of a standard in high throughput drug discovery, especially of antibodies and biologic therapies, to use this. But there's opportunity to use these approaches in more downstream assays. And that's what I tried to focus on today in this talk, is where are the opportunities to use this a little bit downstream in terms of optimization, even things like cell line development or formulation development. So we'll talk through some assays that are relevant for those characterizations and where using some of the throughput can still have some advantages. So the first one is pretty obvious, affinity. Do your engineered candidates, conjugates, process samples bind with the expected kinetics? That's pretty straightforward. The next one is specificity. The binding specificity and relative affinity of biologics and their engineered variants is really crucial for their success. And HT-SPR really enables a rapid and rich characterization of both binding specificity as well as less targeted polyreactivity. Um, you can measure what we call specific activity. That is how much of your protein in the tube actually binds to the target of interest. Um, that can be used for sort of process quality control. It can also be used for stoichiometry and valency of binding assessments. Sort of the question, are all the binding sites expected, available, and active? And then we'll look at quantitation, how much of your product is present. If you're doing uh, transfections or cell line development, you can look at lots of samples very rapidly. And then also using the platform for sandwiching pair assessment if you are building immunoassays. We'll go through all those examples. So a quick background on how these systems operate. So again, this is sort of classical SPR that's been around for a long time, but we bring to it a new fluidic approach. So the platforms all have two semi-independent fluidic modules within them. One is a 96 channel side that flows 96 samples at a time over the surface. This can be robotically moved to index locations. So on the LSA, you have access to 4 nested locations and can create a 384 spot array within the system. On the Ultra, there are 2 locations or a 192 spot array. So the 96 channel side can move off of the surface, clearing the way for a single channel side to come on and do what we call one-on-many analysis. So this is injecting one sample over the entire array. And these 2 systems are robotically controlled so that within an assay, they can come on and off the chip as is needed by the workflow. I have an animation to kind of show this process a little bit. I can get it to play. We call this continuous flow microspotting. So this is the 96 channel side where we have 96 needles descending into either a 96 or a 384 well plate. It docks onto the chip surface, flows the sample back and forth across the chip surface before returning it to the plate. So because the flow is cycling, you can have extended contact times from that small sample volume with no trade-off in flow rate and sample volume. So if you want to capture a low concentration sample for 40 minutes onto the surface, you can do that. Sorry, play it one more time. Oh, okay. After it loads a sample, it returns it to the plate and where it will then rinse the needles and go back and get more samples and address additional array locations. On the other side, we have the single flow cell. And this is pretty straightforward. There's a single needle that accesses a separate rack and plate and draws one sample that it flows over the entire chip surface. So this is a 270 microliter volume sample giving you binding access to the entire chip. So if this is kinetics, that's a single concentration of your target, or single volume of each concentration of your target being injected over. Or if it's a competition study, it can be, one injection volume of competitor giving you information to everything that's immobilized on the array. So we can use that to look at things like capture kinetics and specific activity. So what is capture kinetics? This is an experiment where we have a chip surface that has some intrinsic affinity to the protein of interest. So if it's an antibody, it may be an anti-FC antibody or a protein AG surface. If it's some other protein, it could be anti-His or anti-V5 tag or whatever. So here you can see the way that the experiment works is we're using the 96 channel side to do 3 steps of immobilization. So these are 96 samples loading at a time. You can see this is actually a titration of the same 96 samples 3 different times. Then we do a series of buffer injections to flatten the baseline, and then an increasing concentration series of the target of interest. So this is where we get our binding kinetics from. Because we're using the 96 channel to load samples to an affinity surface and recording that data in real time, you can actually incorporate quantitation with this assay if you included a standard curve in your loading samples. So how do we set these experiments up? So this is a view of the Navigator software in the Capture Kinetics wizard. So we have the sample deck of the instrument shown here, the 3 bays on the right with these 384 well plates. Those are the ligands that are captured by the 96 channel side. And then these are the tubes and plate that are accessed by the single channel side. Um, so in this case, we can create what we call capture sets. So we define a set that includes the printing in for the capture information. So this is 4 steps using each of the 496 quadrants, 96 well quadrants of the 384 well plate. So it's building the array, then it's doing single flow cell injections first of some buffer wells, and then of a titration series of protein. So uh, in the example I'll show in 2 slides, we're doing full kinetics of a bunch of samples, and each one of these titrations only used 5 micrograms of protein. So this is a great way to get really rich, large-scale kinetic screening from commercial sources of antigen. You don't need to buy a huge amount. So if you had your acroproteins, you would titrate them here in these wells and end up with 1,152 kinetics from this experiment. This would run in about 24 hours. Another page of this wizard. So we've got our samples defined, our analyte injections, and here we're setting the time. So we're going to capture these for, say, 10 minutes. Then we do buffer flow, sample injection for the association, and more buffer flow for dissociation. It will loop through these steps as it goes through all of the steps within one of these captures as we've built the array. And then when it gets to the end of that sample list from the previous slide, it will perform whatever regeneration is defined. And then it can sort of repeat the cycle for the next plate or the next target if you want to recapture the same samples. Okay, so this is an example of a 288 interaction array. This was Fabs expressed in periplasmic extracts captured with an anti-CH1 molecule. You can see we get robust kinetics, reproducible. There's 3 prints of the same thing here. That's why the pattern repeats like that. We get pretty diverse kinetics in this set, and almost everything is 1-to-1, fitting that 1-to-1 model really well. So this analysis gives you the on rate, the off rate, the affinity, and an Rmax value, which is that binding level at saturation as determined by the kinetic fit, which is used to do the specific activity calculation. Again, this can scale to 384 well plates in an unattended run in about 24 hours. And this actually is captured in our Fab application note, if you're interested on our website. And it's important to note that getting good kinetics in this type of experiment is really highly dependent on having quality protein used for the injections. If you want to see a 1-to-1 kinetic profile, whatever your antibodies or biologics are interacting with needs to be present in a relatively homogeneous fashion. And so, good sources of protein like acro are ideal for this kind of behavior. And because it doesn't use a huge amount of protein, it's a perfectly appropriate source for this type of analysis, even for large screening campaigns. So to actually do the specific activity calculation, we're now going to compare the amount of ligand immobilized, in this case, the Fab that was captured to the chip surface, and compare it to the Rmax from that kinetic fit. And that result will be presented as this percent specific activity value in the kinetics table within the software. And this can be a key quality attribute. You may have purification fractions or media or fermentation experiments where you're doing that could affect the activity of the molecules you're looking at, or even conjugations or stability studies. So it's a good way to track that activity rapidly and for many samples. So to calculate this percent specific activity, you take the observed Rmax, or the one calculated in the kinetic fit, and divide it by a theoretical Rmax, and then just times 100. The theoretical Rmax is basically just a mass-based stoichiometry signal assessment, because SPR signal is generally tied to mass very closely, especially for proteins. So you have the molecular weight of your analyte times the ligand valency divided by the molecular weight of the ligand times the immobilized ligand level. So it's just basically a stoichiometry calculation of how much you would expect to see binding. To do the determination, when you're looking at the data, there's a report point page where you can define a region that you want to get as your numerical report point. In this case, it's the RUs of the protein captured at that step. Then on another page, you will enter in the molecular weights of your ligand and analyte, as well as the valency of the ligand. Then on the kinetics page, which is shown here, you just check this box that says show specific activity, and it will add a column to the table where it's looking at this our max value. So here we have our on rates, our off rates, our affinities, the KDs, and the R max value from that fit for each one. And it will compare that to the capture level and calculate the specific activity. So you can do this for, many samples simultaneously. And so, we've do a lot of work on development or early screening where there is real high throughput, but in later stage testing, a lot of times there's less unique samples, but that doesn't necessarily decrease the value of the array because you can increase your N and get really good statistical confidence. If you have several fractions or mutants or things you're looking at, and you spot them at several densities at 12 replicates each, for example, you can get a very good sense of how comparable these are and some really meaningful statistics around what is the variation in your measurement in terms of fit. And if you're looking at these things that are like, say, single amino acid mutations in the same flow cell together, getting the same analytes injected over them at a high N, you get the comparability is excellent, and you get a really good estimation of how much variance there is. So the software will actually automatically calculate for you the mean and standard deviation of rate constants and R max values in those if they have the same ligand name. There's a statistics page that will do this. So this becomes a really powerful tool and very sensitive at detecting minor differences, much better than just looking at, say, the standard error of a fit from a lower throughput estimate. Okay. The next thing we'll talk about is quantitation. So we already talked about how you can see the 96 channel loading these samples onto the surface. And if you include a standard curve, you can just generate a 5-parameter logistic fit to back extrapolate the concentrations of your unknowns. And this can scale up to 1,152 samples, including the standards in that. And because it uses the same report point process as the capture kinetics, this could actually be part of a capture kinetics experiment. So when we're looking at the application of the quantitation, you can see here is an example of antibodies being captured to a surface. And we have two regions highlighted here. The beginning of the binding, where for high concentration samples, we see a very steep slope and good separation of the higher concentrations. And then a later curve where we see some binding from the really low concentrations and maybe a bit of convergence on the high. So the standard curve here is shown from the early slope phase, where you can see we're still getting the red dots are the standards and the black are the extrapolated unknowns overlaid on the curve. We're getting good dynamic responses all the way out to 100 micrograms per mil. If you look on the lower curve where we're using the end of injection, there is some convergence of those there, so there's a little bit less dynamic response, but we're actually gaining quite a bit of range on the bottom end. So there's a lot of information contained in these sensograms, and applying the appropriate, report point and standard curve can affect your dynamic range, or you can even run this analysis, say, in both these cases and get a very expanded dynamic range to assess the proteins. So the next thing I'm going to talk about is specificity and polyreactivity assessment. So obviously the specificity of a drug is critical for success, and some polyspecificity can be desirable. You may want a molecule that can neutralize proteins of different species or different isoforms or family members to increase its efficacy. But unintended polyspecificity can decrease efficacy or increase toxicity. You don't want to have off-target binding driving other biologies that are unintended. Polyreactivity, or what is typically thought of as non-specific binding, is this tendency for binding to common chemical features like hydrophobic pockets or charged domains, and is generally a negative for drugs and biologics in general. So candidates with polyreactive profiles will demonstrate poor pharmacokinetics, frequent toxicities, and usually clinical failure. So high throughput SPR is a way you can really rapidly characterize the binding to many targets and assess these properties. So there's a couple different modes you can do a specificity assessment in. One is to immobilize or capture your array of candidates and then inject analytes of interest. Um, Another one would be to capture or immobilize targets of interest and then inject your candidates as analytes. But either of these approaches can really be scaled quite large with automated matrices up to 384 by 384 possible on the LSA platforms. We have two application notes that talk about this approach. Well, one is an application note, the one shown here, the profiling antibody polyreactivity and polyspecificity. Um, there's also a recent peer-reviewed publication that came out of the Journal of Immunological Methods in a collaboration with us and Specifica. And that's actually the example I'm going to show on the next couple of slides. So in this case, we took a series of biotinylated polyreactivity reagents. You can see they're shown here, some albumins, a fibrinogen, ganglioside, polyglutamic acid, and a random DNA monomer. And those were biotinylated, captured onto a Nutravidin chip in a serial dilution. So we had 6 concentrations of each of these reagents, making some different surface densities, so we can see how that changes the activity. And then those were actually spotted. So the whole plate with duplicate titrations was spotted twice onto the array. So we had quadruplicates of every concentration of all of these ligands. Then we went and injected MABs at 250 nanomolar and 2.5 micromolar to look for polyspecificity of the binding. This is an overlay of that data. So you can see we have several different densities or replicates of each of the targets, and the binding response is shown overlaid. So different antibodies that had polyreactivity profile are shown in different colors. So the Lenzi is the blue, so you can see it bound a little bit to BSA, a lot to the monomer and the polyglutamic acid, and gantenerumab bound heavily to fibrinogen and ovalbumin. And all of the others were fairly clean, especially when viewed at this scale. So another way of looking at that data, it would be these box plots where we can see the replicates of the few highest concentrations of each ligand with error bars. And so some targets had sort of low levels of binding, whereas some of the clones bound above background, but the RU response is fairly low. Others had really high response, and so high that it makes it actually hard to see sort of more subtle effects on the same scale. But we can zoom in on that. Like, so here's the fibrinogen, which had the gantenerumab binding at hundreds of RUs. And when you exclude that or rescale away from that, you can see that there are actually other antibodies too that have sort of above background levels of polyreactivity that are probably meaningful and concern and should be considered a flagging activity. So this study showed that the random DNA monomer, the polyglutamic acid, and the fibrinogen were really the most informative of this set of ligands in terms of predicting broad polyreactivity, and also that the 2.5 micromolar injection of the antibody really seemed to give an appropriate sensitivity of polyreactivity. This assay was comparing a similar approach using ELISA, and you could see some binding in ELISA of things that were harder to see by SPR, but it seems like most likely those are less relevant in terms of having any sort of biological effect, because drugs that are showing clinical success and good toxicity and relatively good PK profiles are sometimes flagged in that assay, whereas it seemed like the flagging done in this format at these sort of concentrations was really relevant to predict problematic polyreactivity. So real quick, a couple examples here of additional types of specificity screening. With the Ultra coming out, we're looking more at small molecule binding, and many small molecules bind to a number of targets, and it's often a challenge to do high throughput specificity assessment. So here's an example of a screen we did where we took the Maybridge 1000 fragments, so these are those small molecule binding fragments library, and screened it against 125 biotinylated kinases immobilized to a single array, and selected binders and moved some of those into KD determination. So this screen, which took about 3 days to run, yielded 125,000 interactions. And if you look at this animation on the right, where we see these fragments appearing in the binding pocket of a kinase, if you consider all of the 1,000 fragments against 125 kinases, this is the actual speed at which that screen was occurring. Each injection takes 3.5 minutes, but if you take in the parallelization, so it's pretty phenomenal how quickly you can move through these interactions. Here's the conditions of that. It was a 300 micromolar compound screen. And what you get out of that is basically 125 of these plots where you have each protein against each fragment. We can see our ADP controls, and then we can flag clones that bind above background. And there's statistical tools to use this. You can also obviously see their sensograms. Fragments tend to bind very rapidly, so they tend to give these square profiles, at least if it is a specific interaction. Extending that, you can do things like kinase profiling. So again, similar uh, array where we have, I think in this case it was 87 kinases, active kinases immobilized, and we did kinetic titrations of different kinase inhibitors. And so in a single experiment now, basically in a day, you can get uh, activity profiling of that molecule against whole families of kinases. And this type of detailed analysis really would have been prohibitive on any other type of platform, at least to get real-time kinetic binding. Okay, so so oftentimes you need good sandwiching pairs to do biomarker analysis, anti-idiotype PK/PD studies, or looking for things like infectious diseases in serums. So the HT-SPR is really a fantastic way to assess those reagents. You can immobilize them onto the array in mass and do detailed kinetic analysis, figure out which are your highest affinity, which ones bind the most rapid, which ones bind the most stably. You can do things like specificity testing, whether or not they bind to drug, pro-drug, conjugate, not conjugated, free drug, forms like that very rapidly and easily with a rich picture. And then you can do fully symmetrical pairwise competition-based epitope binning, which will allow you to see which clones form effective sandwiching pairs. And because the data is real time, you get these binding sensograms you can look at. So you don't just know that they sandwich well with each other. You can see how rapidly does the complex form and how stable is it in sort of a real-time visualization. So this can really inform the selection of which reagents are worth doing whatever labeling you need to do to make your final format for your actual sandwich assay, to do the testing. So you can go from having many potential pairs and orientations to test to just a few that are very likely to be successful. And it's not always obvious, which will be the best capture and detection, but typically this assay format will give you that information ahead of time. Okay. And then the last example I'm going to talk about here is not so much of a late stage development, but it is one of the things that really highlights, what you can do in drug discovery and development if you have all the right tools in place. And this is the Bamlanivimab story. So when COVID-19 broke out, Eli Lilly and AbCellera had a pre-existing arrangement with DARPA for pandemic readiness. And AbCellera, so both of these were Carterra customers with platforms in their labs, or LSA platforms in their labs. And AbCellera in Vancouver, British Columbia, got blood from one of the first COVID recovered patients in Canada and did a B cell discovery effort to find putative B cell spike protein binders. And assess those in their in silico system to identify, okay, what are interesting sequences that seem diverse and likely to be potential neutralizers, and started expressing those. So in the first week, they made 187 clones and shared those with Eli Lilly. And so those were all processed for affinity to spike and RBD. They were run in pairwise competition-based epitope binning to figure out what was the epitope diversity within that, and biochemical ACE2 neutralization. So from that, they were able to assess what were high affinity neutralizers from a diverse set of epitopes. And they picked about 24 clones to go into developability and potency downstream work. And from this initial run through, one of the clones became Bamlanivimab and was actually had was in the clinic. I think it was 94 days after the initially getting the blood from those patients. So just a phenomenal speed. And what was interesting too is that as the SARS-CoV-2 evolved and the bamlanivimab stopped working against the later mutants as it was generated against, the very first strains of COVID-19 that came out, they were able to go back to this B-cell sequence repertoire and find, okay, what were rare epitope bins that showed resistance to viral escape or viral mutation, and mine in, and they actually came up with beptulivimab, which was a follow-on approval, from the same patient samples, and that one was effective all the way through. I think it was the Omicron 1.1 strains, when eventually viral mutations decrease that activity. So we think this is a great story about, if you have the right types of platforms in place to really get rich information early on, how quickly some of these things can move through development. And with that, I want to thank the Carterra team, especially Rebecca Rich, who did most of the small molecule work. I showed collaborators like Adam Schutz and the folks at Specifica, which we did the polyspecificity work with. And the Fab binding work I showed was in collaboration with Christopher Brandstrom at Pfizer San Diego. And again, thank you to Acrobiosystems for being a great partner with us and allowing us to talk today. So thank you.
Thank you so much, Dan. Are there any questions for Dan? If so, please feel free to raise your hand or just type in a chat box. I do have one question on the sensitivity range. What is a typical sensitivity range you see for the quantitations?
Yeah, so it is going to depend somewhat on what type of capture surface you're using, but typically for an IgG with a high affinity capture surface, it's going to be in the low mid double digit nanogram per mil, so say 30 nanogram per mil to about 100 micrograms per mil. As proteins get smaller, the molarity uh, goes up per mass. So you probably could go lower in the nanogram per mil range if you have a high affinity capture, but it's kind of broad range. So you usually get, a good 3 logs of dynamic range.
Gotcha. Thank you. I don't see more questions. But also, I'll just ask another question. But when in
Thanks.
the selection and characterizing process, when is it appropriate to actually look at the reactivity of the candidates?
Sure. There's a lot of different ways you can do that. And we have customers that start addressing polyreactivity concerns as early as like the early primary screen. So if you're running, say, a capture kinetics from crude samples, you may want to inject 2 or 3 of these, high value polyspecificity reagents over the captured array to look, sort of immediately early on for sort of gross effects. But then you can also do it later on downstream when you have a better sense of your proteins and they're purified at high concentrations. You could do a more rigorous screen on panels of immobilized polyreactivity. So I think there's opportunity to do it sort of throughout the process. Earlier is probably better. It's Not something you probably want to spend a lot of time engineering away from your proteins as if they're
Yeah,
sticking to polyreactive surface. So I think, addressing it as early as possible is probably to your advantage.
gotcha. Thank you. And I see two questions in the chat box now. Oh, wait, three. And the first one is, how many times can the sensor chips be reused? As in like docked and redocked?
It depends on the sensor chip. Some things like nickel NTA and protein AG are probably more robust.
Mhm.
The surfaces themselves a lot of times can be regenerated many times. I, when you remove a chip from the system and put it back in, what you need to try to preserve is the optical quality of the chip surface because it's sort of a large, flat, highly optically, sensitive surface. And what we see is it's stored in the fridge. A lot of times the chips just get messy looking. Like if you look at the image of them, they will get features on them. So we found that doing some things like storing them with 50% glycerol on the surface preserves them better. And you should be able to get them in and out if they're a capture surface, in and out of the fridge a few times. Um, Not indefinitely, but it depends a little bit on how they're handled and, how much buffer sort of dried on them when you were using them, things like that. So several times, not many.
Yeah.
But they can often be regenerated many times. Like if you have a capture surface, you can have it in the instrument and regenerate it 20 times or more, and it will be fine.
Gotcha. Uh, we have three more questions, but feel free to also type it afterwards. I'll
Yeah.
go through one more since no, the time is up almost. It says, are there any specific tools and epitopes to help identify the best sandwiching pairs?
So the normalized response value that is sort of the value in the heat map and epitope is an indicator of how large the sandwiching signal is. So I think, all the tools in epitope will tell you kind of, the heat map and the network plots tell you whether things do sandwich. I think to actually find the best ones, you might want to actually look at the sensogram. So there is an array view where you can look at many sensorgrams at once, and ones that, form very rapidly and stay high and look very stable are often good ones to look at. So, it's pretty easy to see them. But yeah, I guess the value in the heat map would probably be a good starting point.